students, welcome back to Kingdom Zoology. Let's continue our biotechnology lessons. We have discussed in biotechnology till now what exactly is the purpose of biotechnology. That is basically producing a product of human interest. For example, any protein product that you actually require to control or treat diseases in the humans. We have to produce that protein product at mass scale. That's what biotechnology is all about. So for this, what we have done till now is identify the cell of an organism that is the natural producer of your desired product. Once you have identified the cell that is the natural producer of your desired product, you have to take out the genome of this cell. And once you have taken out the genome, the next step that we discussed was trying to cut this genome into smaller fragments or pieces. From those pieces, one of them would be actually coding for your desired product, right? So now we have discussed over here that when you want to cut the genome, one option that you had was exonuclease enzymes, like the ones which are found in the human pancreas or the human digestive system. But the problem with those enzymes is they begin from the ends of a DNA and keep cutting every single phosphodiester bond, ultimately producing individual nucleotides that will be of no use because they are not going to code for any product. So if I want to cut the genome and produce various gene fragments from it, maybe cut it over here, cut it over here, cut it over here, so that you can actually get what? Different gene fragments. One of these fragments may be having the property to code for your protein product. So that is the fragment which will be called desired fragment. So to obtain the desired fragment from the genome of this cell, what I need are enzymes called restriction endonucleases. The history of restriction endonucleases we have discussed already. They are enzymes that would be obtained from bacteria. Why do the bacteria have them? As a natural defense mechanism against bacteriophage viruses. So we can obtain these enzymes from the bacteria, use them in biotechnology to cut down this genome. Once the restriction endonuclease is done with its action, the genome would have been broken into many pieces, and you just have to identify the correct piece that would be coding for your protein product. Now, before we actually move to that part where we have to cut the genome into fragments and then identify the correct fragment, we must have a look at different types of restriction endonucleases available to us. So that is what this lecture is all about, various types of restriction endonucleases, which have been isolated from bacteria for use in biotechnology to cut a genome and produce various gene fragments with it. So in the types of restriction endonucleases, we have three choices. Number one, or we can actually write them as A, B, and C. Type 1 restriction endonucleases. If you want clarity on why we are using restriction endonucleases and why are they called restriction endonucleases, please watch the other video where we have discussed about digestion of the genome under the biotechnology playlist. So type 1 restriction endonucleases. All the restriction endonucleases are obtained from bacteria only because that is where they are naturally found. So type 1 restriction endonucleases are the ones that identify a particular palindrome. Palindrome, I hope you can recall what it means. A type of sequence in the DNA that actually has same base pairs from both the sides. For example, if this is your DNA strand with the nucleotides on it, over here is the 3' end, the second strand 5' end, and this is your, let's say, the 3' end. So in this DNA sequence, let's say there is a small area with this particular sequence. So when you look at the reading frame, it's GAA-TTC from 5' end to the 3' end in this direction, and it's same GAA-TTC from 5' to 3' direction in the other strand also. So such DNA sequences are called palindromes. So type 1 restriction endonucleases are the ones that would identify a particular palindrome on the genome to be digested. But the problem with these enzymes is once they attach to this palindrome, they are not going to cut any phosphodiester bonds within the palindrome. The enzyme is going to shift, and the shifting direction is also not fixed. They can shift to left or they can actually shift to right side of the site of attachment, and they can actually move as long as 1000 base pairs away from their recognition site and cut at a random place anywhere. They will attach to a particular palindrome on the genome to be digested and then move or shift approximately 1000 base pairs away from the recognition site. This is basically to cut the phosphodiester bond. So we can notice that recognition site is a palindrome, but where exactly would they cut? Is that site also fixed in them? No. They can shift to left or right of the recognition site and introduce random cuts at any place on the phosphodiester bonds very far away from the recognition site. This type of enzymes are called type 1 restriction endonucleases. We'll also discuss about why this is a problem, not cutting on the recognition site. So we'll have a discussion over it also in a while. Before that, let's write down one more property about these enzymes. These enzymes require magnesium ions and ATP as their cofactors. Cofactors in case of enzymes are the non-proteinaceous parts. Enzymes can be divided into simple enzymes, as we know from biomolecules, or conjugate enzymes. Simple enzymes are the ones that are made up of only protein parts. Conjugate enzymes have got protein part and the non-protein part. The non-protein parts of conjugate enzymes are called cofactors. So magnesium and ATP are the cofactors for the activity of type 1 restriction endonucleases. The next type of restriction endonucleases are type 2 restriction endonucleases. What is special about these enzymes? They also identify a palindrome. The palindrome can also be called the recognition site of the enzyme because that is how they recognize the genome on which they have to introduce a cut in the phosphodiester bonding. So type 2 restriction endonucleases also identify a palindrome recognition site. And an important point, they cut a particular phosphodiester bond within this recognition site. So what is important about them is they are going to identify a particular palindrome, and once they have identified a palindrome, they are going to cut the phosphodiester bond within that palindrome. Look at this again. If this is your genome that needs to be digested, the enzyme is going to attach to this site, and then once it attaches to this site, imagine it's going to cut the phosphodiester bond between G and A on both the strands. So as a result, your genome gets broken into two pieces now. The two pieces will be these. This is one fragment. This is the second fragment. So we have broken or digested the genome into fragment number 1 and the fragment number 2 because of the cuts on the phosphodiester bond introduced by type 2 restriction endonucleases. So what is fixed about them? Both the things, recognition site as well as the cutting site. Plus, the important thing about these enzymes, they require only magnesium ions as a cofactor. There is no requirement of ATP for the action of these enzymes. Require only magnesium ions as cofactors. The third type of restriction endonucleases are type 3 restriction endonucleases. Same, identify a palindrome. So that part will be same for every single restriction endonuclease. They also identify a palindrome, that is the recognition site, and cut, not exactly within the palindrome, but not exactly very far also, around 25 to 27 base pairs away. But then away means again in either direction. The enzyme attaches over here, shifts this side or this side, 20 to 20, 25 to 27 base pairs away, and introduces a cut in the phosphodiester bond sequence. Identify a palindrome and cut 25 to 27 base pairs away from the identification or the recognition site. So these enzymes are called type 3 restriction endonucleases, and just like type 1 endonucleases, they also require magnesium ions and ATP as their cofactors. So see, the situation is there is only one type of enzyme out of these three that is identifying a palindrome and cutting the phosphodiester bonds within that palindrome. The other two types of enzymes, type 1 and type 3, they will be identifying a palindrome. So the identification site for them is fixed. But where exactly would they introduce cuts? That is not fixed for the type 3 and type 1 enzymes. So look at this now from the perspective of digestion of the genome. Here's your genome that needs to be digested or broken into gene fragments. This is the same genome that we had obtained from the cell, who is the natural producer of our desired product. But then that cell produces the desired product only for its own purposes in low quantity. We want our desired product in mass quantity. That's why we are doing all this. So here's the genome that is extracted from the cells who are the natural sources of our desired product. But the genome, the same genome that was actually isolated with the help of or precipitated with the help of absolute chilled ethanol. So I have to digest this genome, and to digest this genome, I can actually add restriction endonucleases where I have three choices: type 1, type 2, type 3. The problem with using these enzymes is they will be identifying a particular recognition site on the palindrome and will attach to it. But what type of fragments will they produce? Random fragments, because we have no idea where they are going to cut. Let's imagine a situation where this is your genome, the one that I wanted to digest with the help of the restriction endonucleases. Let's imagine a situation where I added type 1 or type 3 restriction endonuclease. It identified a particular palindromic site on the genome and then shifted 1,000 base pairs away, or maybe 20 to 25 base pairs away on either side, depending on which enzyme it is. So let's say this is your attachment site of a type 1 or type 3 restriction endonuclease. Maybe let's say I'm using type 3. So when the type 3 enzyme attaches over here, it is going to shift on this side approximately 25 to 27 base pairs away and maybe introduce a cut over here. So that is okay, not a problem. What do I get? Fragment number 1 and fragment number 2. These are the two fragments that I've got. And let's imagine this is the fragment which actually has the property for coding for the desired protein product. So as you can notice, there is no problem then in using the type 3 enzyme. I have the genome, I can add a type 3 or a type 1 enzyme for that matter, and the enzyme is going to identify a palindrome. This is the palindromic identification site. Then the enzyme shifts maybe to the right side around, let's say, 26 base pairs away, introduces a cut over here at this place, produces fragment number 1, fragment number 2 from this genome, and this fragment number 1 has got the property for coding for the desired protein product. I've got the desired protein product, done and dusted, मेरा काम हो गया. तो टाइप 3 एंजाइम यूज करने में भी कोई प्रॉब्लम नहीं है. बट प्रॉब्लम कहाँ पे आएगी? रिपीटिशन में. When you want to repeat the same experiment to get the same fragment so that you can get more protein product, you have to repeat, right? To get more product, you need more copies of this fragment. And to produce more copies of this fragment, you'll have to repeat the process, genome isolation and digestion with the same type of enzyme, type 3. So let's say again, I get the same genome and I put the same enzyme, type 3, which I had put last time. This time also what will be fixed? The recognition site. The enzyme is going to attach to the same recognition site. But the problem is, let's say the enzyme shifts again on the right side, but this time it actually moves a bit far ahead and introduces the cut at this location rather than where it did the last time. So now if the cutting site has shifted, can you notice that the gene fragment that we were talking about, the fragment that was basically coding for my desired protein product, that fragment itself has been cut through the center now, or cut in between now. So the desired gene fragment that you got in the previous attempt, you are not going to get the same, or there is no guarantee that you're going to get the same fragment. It's quite possible that the enzyme may introduce a cut within the desired gene fragment this time. Because what is fixed about type 1 and type 3 enzymes? Only the recognition side. What is not fixed about them? The cutting side. So when they identify a genome and introduce a cut at a particular place, producing fragment number 1 and fragment number 2, and let's say this is the fragment that is coding for your desired protein product, there's no guarantee that the same enzyme is going to produce the same fragment next time. You take the same genome, add the same enzyme once again, but there's no guarantee that you're going to get this product or this gene fragment again. It's possible that the enzyme would move a bit forward and cut next time at this place. If the enzyme cuts next time at this place, it has now digested your desired gene fragment itself into what? Two pieces. So last time you cut the genome by using a type 1 or type 3 enzyme and you got a desired gene fragment that coded for the protein product. If you want to repeat the experiment, produce the same protein product again by cutting the same genome with the same enzyme, will that be possible? Maybe not. So there's no point in using such enzymes. Then we should be actually using those enzymes that would be identifying a particular palindrome on the genome, cut within the palindrome and produce fragment number 1, fragment number 2. Let's say fragment number 1 is the fragment that is going to code for your desired protein product. So this is my desired fragment. If I want more copies of this desired fragment, I can actually isolate more genome from the same cell by the same method and add the same type 2 restriction endonucleases. If I'm using the type 2 restriction endonuclease, same, the one that I used last time to cut this genome into fragments, is it guaranteed that the fragments that I got last time, I'll get the same fragments this time also? Yes, that is the guarantee in type 2 restriction endonucleases, because they are going to identify a palindrome, and once they identify a palindrome, they're going to cut within the palindrome every time at a fixed place. So the type of fragments that they produced in the first attempt of cutting the genome, the same fragments will be obtained in the second, third, fourth, fifth, any repetition attempt. So what enzymes, as you can notice, work for us in biotechnology? Type 2. Okay. So if this is a genome to be digested or broken into gene fragments, we can add type 1, 2, or 3 restriction endonucleases, and all of them will digest the genome into pieces or gene fragments by attaching to the genome on the recognition sites or palindromes and cutting wherever they want, within the palindromic site, far from it, close to it. So it doesn't matter. You can add any enzyme and they will basically produce fragments. And one of these gene fragments that you have got over here by the digestion of the genome will be capable of coding for our desired protein product, which we want to produce in mass scale. And that's why we are doing all these efforts. One of these fragments will be coding for our desired protein product. So that is okay. In the first attempt, there's no problem. You can use any enzyme, cut the genome, produce fragments, and one of those fragments will be coding for your desired protein product. The problem arises when you want to repeat. If we repeat the genome digestion process, extract fresh genome, put the same enzyme again. If we repeat the genome digestion process and add the same restriction endonuclease that we used in the first attempt of cutting this genome into fragments, we will be getting the exact same gene fragments as obtained in the first attempt. Because we want the exact same gene fragments, because one of these gene fragments was coding for my desired protein product and I want more copies of that gene fragment. So I'll be getting the exact same gene fragments as obtained in the first attempt. तो बताओ किसमें? Only if we use what type of restriction endonuclease? Type 1, type 2, or type 3? If you just thought of type 2, that is the right answer. Because if I had added type 1 or type 3 enzymes in the previous attempt, even then I would have got gene fragments and maybe one of those coded for the desired protein product. But if I want to cut the same genome with the same enzyme and expect the same fragments next time also, it's not necessary that I'll be getting the same fragments because the enzyme may cut at a different place this time. But with type 2, that problem is not there. Type 2 enzymes will identify a palindrome, cut at a particular place, produce particular gene fragments. One of those gene fragments will be coding for your desired protein product. Take the same genome again, put the same type 2 enzyme again, you'll get exactly same gene fragments the second time also. So this brings us to the conclusion that the best enzymes for genome digestion in biotechnology, in modern biotechnology or genetic engineering process are type 2 restriction endonucleases. So that's it, guys. This explains our differences between the type 1, type 2, type 3 restriction endonucleases, the choices available for digestion of genome, what exo and endonucleases, what did we prefer, endo, what type of endonucleases. We had three options, type 1, type 2, type 3. What did I choose? Type 2 on the basis of its property or capability of producing exactly same gene fragments every time I digest a genome by using them. Okay, so I hope you understood about type 2 restriction endonucleases and why we use them. In the next lecture, we'll continue the story forward where I'll explain to you about two different options within type 2 restriction endonucleases. Those two different options are blunt end producing type 2 restriction endonucleases and sticky end producing type 2 restriction endonucleases. For that, stay tuned. I'll be uploading another video. Till then, keep studying, keep sharing about the channel, keep liking the videos. Thank you so much.